For decades, historians modeled the Viking Age as a simple demographic expansion, a homogeneous Scandinavian population sweeping outward across Europe. In 2020, researchers from the University of Copenhagen challenged that assumption. They published a study in Nature analyzing the genomes of 442 ancient human remains. By utilizing shotgun sequencing on these remains to a median depth of 1x, the team isolated specific allele frequencies across the population. The resulting data proves that the Viking was not a distinct biological group. Instead, the sequencing reveals a heterogeneous genetic mosaic. Biology and history separate here. The data forces us to define Viking strictly as an occupational title and a cultural matrix, rather than a shared genetic heritage. The sequenced samples cover a vast geographic scale, yielding DNA from remains buried as far east as the Asian steppes and as far west as the American continent. To accurately map this world, we have to abandon modern political borders entirely. The structure of the Viking era only emerges through bioinformatic models and data clustering. To detect fine-scale population structure among these ancient samples, researchers utilized a statistical tool known as identity by descent, or IBD, segment mapping. This UMAP plot organizes hundreds of genomic data points into 3 separated coastal clusters: Danish-like, Swedish-like, and Norwegian-like. Mapped against topography, these align perfectly with fjords and mountains. In southwestern Sweden, the population belonged to the Danish cluster because inland mountains blocked gene flow. Physical geography and maritime access dictated the genetic structure of the Viking heartland. A contrast emerges when comparing coastal trading hubs like Gotland and Skara with surrounding territories. The coastlines exhibit significant genetic diversity. Meanwhile, contemporary agrarian communities located just inland were isolated. They show no genomic admixture from the preceding Iron and Bronze Ages. Utilizing F4 statistics, researchers proved that the Viking Age featured heavy bidirectional gene flow. It was not a one-way outward expansion. Southern European and Asian ancestry flowed directly into these Scandinavian coastal centers. A coastal Viking living in Skara was biologically closer to a foreign trading partner than to a farmer living a few miles inland. Researchers tracked where these specific coastal ancestries expanded over time by using chromopainter and spatial interpolation models. This interpolation map shows Norwegian-like ancestry flowing westward across the Atlantic into Ireland, Iceland, and Greenland. The Swedish-like component followed an eastward trajectory, moving into the Baltics and the Asian steppe. Finally, the Danish-like ancestry traced a primary vector south into the British Isles. These groups maintained isolation abroad. Danish, Swedish, and Norwegian populations rarely intermixed during these early maritime expeditions. The Viking expansion occurred as a partitioned series of distinct, topographically driven maritime vectors. The famous Salme ship burial in Estonia provides a fascinating microgenomic case study of an early Viking raid. This pedigree reconstruction chart utilizes Primus and Reed kinship coefficients to highlight direct familial links between 4 specific male individuals buried side by side. The sequencing confirms these individuals possessed Swedish-like ancestry. This specific expedition was a homogeneous familial raiding party. Contrast that with Viking burials found in Orkney, Scotland. The individuals in these graves were interred with high-status Viking weaponry and cultural rites. Yet genomic sequencing proves the Orkney individuals possessed 0 Scandinavian DNA. They were genetically identical to native Picts. The Viking identity operated as an open cultural framework. It manifested as a biological Swedish family in Estonia and an assimilated Pictish population in Scotland. Alongside population admixture, the region experienced sharp allele frequency shifts. By tracking these changes over a 10,000-year timeframe, researchers identified specific traits driven by positive selection. This line chart tracks the LCT gene allele responsible for lactase persistence. The frequency rises sharply starting in the Bronze Age. It levels off exactly at the Viking Age timeline marker, hitting the high-frequency percentages observed in present-day Northern European populations. Other specific loci associated with complex traits, including pigmentation and the secondary immune response, underwent strong population differentiation during this window. The Viking Age coincided with the stabilization of these traits, establishing the specific biological profile observed in Scandinavia today. The data replaces the model of a pure biological wave with a maritime transport zone connecting distinct coastal hubs while leaving inland agrarian territories isolated. The Viking diaspora was an occupational phenomenon, pulling diverse global DNA into these cosmopolitan coastal funnels. The legacy of this era is preserved in its cultural artifacts, even in regions where the actual Scandinavian genetic footprint remains limited. Genomic sequencing maps a diaspora characterized by trans-regional engagement, revealing a multi-ethnic maritime network that operated across the European continent.